Do you guys remember what action potential is? Do you guys know how many different types of cranial nerves are there? There are 12 pairs. Olfactory, optic, oculomotor, trochlear, trigeminal, abduction, facial, vestibulocochlear, glossopharyngeal, spinal accessory, and hypoglossal. 10th is vagus nerve. Okay. So, what is action potential? Action potential. Did you guys remember what's depolarization, repolarization? What happens in depolarization? In depolarization, sodium gets inside the cell, and in repolarization, potassium comes out of the cell. So sodium from extracellular fluid comes inside the intracellular fluid, and potassium from intracellular fluid goes outside to the extracellular fluid. Okay? Today is about frog's heart. Okay? So what do we know about frog's heart? How many chambers are there? What are they? Right and left atrium, and only one ventricle. Okay? This is the circulation. This is the anatomical diagram. Okay? You don't have to write this, it is already there in your book. This is the sinus venosus. It is situated dorsally to the frog's heart. Okay? At the back side of the heart. This also acts as a stimulation. Just like SA node, it also gives stimulation to the heart for it to contract and relax. Okay? So how are we going to prepare the frog for this experiment? In this experiment, first we will keep the brain of the frog intact, because we need the cranial nerve known as vagus nerve. Okay? For this to happen, we have to destroy the spine of the frog. Okay? Keeping the head intact. After that, we will place the frog in a supine position and dissect it. After that, what we will do? We will take the heart. Suppose this is how the heart is. These are atrium. Above is the atrium. Lower is the ventricle. We will invert it, antivert it. We will pick up the heart upside. Okay? And use pins to place the heart in a place. After that, we will use a hook, but we should notice that we can't penetrate through the ventricle. We have to hook it on the outer layers of the ventricle. Okay? This is Starlin's liver. Okay? How will you distinguish between this and the normal one? You can see there is a pin, spring there. Okay? Now what we will do? We will take that hook, and with the thread we will round it up in this part of the liver. This is your usual kymograph. In this kymograph, we will rotate at the speed of 2.5 millimeter per second. Okay? Only then the waves of the heart, of the frog's heart, will be visible. This is the normal setup, but in this case, we don't need the other part. We only need that stand, that lever, and the frog will be placed in the table with the heart upwards. Okay? This is how the cardiogram occurs. Since the heart is upside down, the cardiogram is like that. When you take out the paper, you have to turn it around to get the actual. These are the waves. When very minutely, when the rotates, kymograph rotates, then only you can see the sinus activity, which is from the sinus venosus, which is placed at the dorsal part of the heart, then the atrial activity, and then the ventricular activity. Since the ventricle is the strongest and biggest chamber, its contraction is more, so the wave formation is bigger. This is how the normal graph will be seen. You won't be able to notice the sinus, this sinus activity. Only atrial activity and ventricular activity. Okay. This is again from that experiment which happens when you give a stimuli which is hot, which is warm, and then stimuli which is cold. As you can see, the first stimulations are normal. When heat is being exposed, it becomes contracted and more waves can be seen. And when we pour the cold solution, Ringer solution, then there is a huge gap between the normal. This is how the normal graph is. Remember, the frog's heart is about 25 beats per minute. Okay, what is the human heartbeat, normal heartbeat? 72. If you are asked what is the normal heartbeat, then you have to say 72. Okay, 72 beats per minute. Now what happens when we give adrenaline to the heart muscle? Then the spikes becomes more and rapidly. Since we all know that adrenaline is like a flight or fight hormone that triggers when we get frightened or when we are excited to do something, then the adrenaline spikes up. So like that, in normal, when it is in normal condition, it is having normal contractions, but after introduction of adrenaline, heart rate spikes. And similarly, acetylcholine decreases the heart waves. This the same as the before, but in this condition, what happens when we introduce calcium chloride? So you all know from the previous experiments, that when calcium is more, fatigue occurs. When the calcium is already in the sarcoplasm, it's not able to relax properly, which is why the contraction remainder increases. So this is how, when calcium is provided, so the contraction, you can see the wave got disappeared from the above part, which means when it was about to systole, which means, what is systole? It is the contraction of the heart. And what is diastole? Relaxation. That means when the calcium is more in the blood or in the muscle cells, then the contraction is not able to relax, which means actin and myosin becomes contracted and the heart is not able to relax. That's why it got stuck in the systolic phase. Okay, and then when it after some time the waves become normal. After that, when we introduce potassium, as we all know in the action potential, for the stimulation to happen, potassium has to get released from the cell. It has to go to the extracellular part from intracellular part. But in this condition, we are giving potassium to the extracellular part, so there is already huge amount of potassium present in the extracellular. So intracellular potassium is unable to go outside. That is when the diastole happens, and from that position the diastole is unable to occur. Okay. So did you guys understood what it means? So in the beginning of the experiment we told that we have to keep the vagus nerve intact because of it is connected to the brain, the cranial part. So now what vagus does, it decreases the heart stimulations. Okay. When we stimulate the vagus nerve, the heart rate decreases, but you can see there are still spikes present. So what happened here? When the vagus nerve has the function to stop the beating of the atria, but it doesn't have any power over the ventricles. So now what happens? The ventricular part on itself, it's contracting. So that is when the vagal, the heartbeat, we can see very less, but it's still there. The heart is still contracting even though it is being stimulated with the help of vagus nerve. So this is what is called vagal escape. Okay. After we stop the vagal stimulation, the cardiac rhythm comes to normal. Okay. This is the actual graph which has been taken in the class. That's it.